Hello friends, in this video we'll study about action potential and its propagation. We have a graph over here which shows time on this axis and this is the stimulus, the intensity of the stimulus. This is the threshold of the stimulus, and when the stimulus is crossing the threshold, it reaches this place where it is known as action potential. So it is showing the characteristic shape of action potential and also reflects changes in membrane potential. Movement of ions through voltage gated sodium and potassium channels cause these changes. First of all, sodium channels will open and the action potential is initiated. As it proceeds further, sodium channels remain open but become inactivated. Now, what is an inactivation loop? In this diagram, see, this is the inactivation loop over here. It is a channel protein which blocks ion flow through open channels. Sodium channels remain inactivated until the membrane returns to the resting potential. Potassium channels, they open slowly and they remain open and functional until the end of action potential. We have a diagram here which is showing the resting state of the membrane. This is the outside of the membrane and this part is the inside of the membrane, and this is the membrane and these are the channels. These are the sodium channels and these are the potassium channels. This is again a sodium channel. While the membrane is in a resting state, we all know that the outside of the membrane is positively charged and the inside is negatively charged when there is no stimulus. At this point, all gated channels are closed. Both sodium and potassium, they are closed, but some potassium channels are open. Resting membrane potential is maintained by ungated channels and they are not shown in the diagram. Okay. Now here, some gated sodium channels, they open when a stimulus depolarizes the membrane. See here, this channel was closed in the above diagram, but here it has opened and sodium is allowed to come in the inside of the cell. Sodium diffuses into the cell and sodium inflow will keep on occurring if the stimulus is strong enough. This will cause further depolarization and more gated sodium channels will open and more sodium diffuses inside. Okay. Now, in this diagram, what is happening? Because of depolarization, most sodium channels open. See, sodium is going inside. Sodium is going inside. So, most of the channels are open. And the potassium channels, they are still closed. Inside of the membrane becomes positive as compared to outside. See, this is changed. Here it was negative, the inside of the membrane. Here it has become positive. So this is depolarization. In this diagram, the action potential is falling. Most of the sodium channels have now become inactivated. You see the inactivation loop. They have become inactivated. Sodium inflow is blocked and the potassium channels are mostly open. These potassium channels, they are open. Okay. Potassium will go outside here. Inside of the cell now becomes negative again. This is the falling phase. Okay. Here, some potassium channels are open and sodium closed. Some potassium channels are open. But some sodium channels are unblocked too. Membrane returns to resting stage. This is undershoot because permeability of membrane is more for potassium than sodium. And at this stage, what is happening? Permeability of membrane is more for potassium than sodium. Why? Because sodium channels have already closed. We can also say that the end of sodium inflow, the sodium inflow is now stopped. This allows potassium outflow and this repolarizes the membrane. This is what is actually happening. Now, what is refractory period? The time period during which a second action potential cannot be initiated on arriving of a depolarizing stimulus is known as refractory period. And this is because the sodium channels are closed during early part of the undershoot and the falling phase. When they will close, no more depolarization can occur. This refractory period is due to closure of sodium channels and not due to any ion concentration gradient. Okay. So if an MCQ is asked, we have to reply that the refractory period is because of the closure of sodium channels and not due to any ion concentration gradient. Now we see how an action potential is conducted, how the action potential is conducted further. Or we can have a diagrammatic view of the same thing which we studied in the channel and the gates method. Here we are showing in a simplified line diagram. This is the plasma membrane, this is the cytosol, and this is the action potential. At this point of membrane, the action potential is arising, and this is allowing the sodium inflow. And here we see the change of the charges. The inside is now become positive and the outside is now negative. So, sodium flow inside the neuron membrane at the site of action potential, this is occurring in this diagram as we saw, this generates an electric current and the nearby regions of the axon membrane are depolarized. Okay. When this crosses the threshold, action potential is generated, yes. These events are repeated along the axon's length. See here, now the action potential has arisen here and the same event which was occurring over here, it is now occurring over here. So this is a repetition of the events. Sodium is going inside and the change of charges has occurred and they finally reach the synaptic terminal. When the depolarization zone is traveling, the sodium channels behind the zone become inactivated. Behind the zone means this side. This side they have become inactivated. Here we see the potassium channel is become activated and the potassium ions, they are coming outside, not the sodium. So this causes that part of membrane temporarily refractory to a new stimulus. This part, the sodium channels have now become closed. So this part is not temporarily refractory to any new stimulus. It won't get activated. So the action potential doesn't travel back. So if any question is asked why action potential is not traveling in the back direction, why it is only propagating to the front direction, because the previous sodium channels have become inactivated. This is the answer. Now, wide axons, they have more conduction speed than narrow ones as they offer lesser resistance. Vertebrate axons have a peculiar adaptation which allows fast conduction speed in spite of narrow diameter. This adaptation is myelination. Myelin sheaths surround vertebrate axons. This sheath is secreted by glia, oligodendrocytes in central nervous system and Schwann cells in peripheral nervous system. Glia wrap axons in many layers which are lipid in nature and they are poor conductors of electricity, or we can say that they are insulators. Have a look at this diagram. This is the axon, this yellow part is the axon and this is the myelin sheath, and if you have a look at the cross section of this thing, we see the layers of myelin. Okay, and this part, this is not insulated, this is open. This part is also open. These open parts, they are known as node of Ranvier. So voltage gated sodium channels are found only at the nodes of Ranvier, which are areas of no myelin sheath. See, here I have shown you these voltage gated channels. Here also the voltage gated channels is green colored, you can see clearly. Okay, so they are found only at these nodes. in myelinated neurons, of course. Therefore, action potential is generated only at the nodes. Only at these areas the action potential is generated because this part is insulated. So, from this node to another node, the action potential will actually jump. And this jumping allows a really fast conduction, and this is known as saltatory conduction. So, this is all about this video. Thank you. Thank you, friends. Thank you for watching.